Today we're covering mass transport in plants and specifically looking at translocation in the phloem. And by the end of this video, you will be able to answer a 5-mark question on the transportation of sugars in plants. So translocation is just the transport of organic molecules, for example, sucrose, amino acids, and some mineral ions. Sugars are made in the chloroplasts of cells during photosynthesis because, as we know, plants are capable of making their own sugars. These sugars then need to be transported around the plant to the places that require it. This is where the phloem comes in. The phloem transports sugars from the source, which is the site of production, aka cells containing chloroplasts for photosynthesis, to the sink, which is just a name we use for the places that the sugars will be used directly or stored for the future. Sinks can be anywhere in the plant, and so the translocation of molecules in the phloem can be either direction, so it needs to be able to go in both directions. So let's take a look at the mechanics of translocation, which actually isn't fully understood by scientists yet. Materials are transported too quickly to be explained by diffusion, and so scientists favour the mass flow hypothesis, which is a theory that you need to know for the exam. The wording mass flow simply refers to the bulk movement of a substance through a given area in a specified time. Before I go into the steps of translocation in plants, I just want to say that when you're talking about sugar and the movement of sugar in this process, mark schemes will allow you to use the word sucrose or sugar, but not glucose. So I would recommend that you go with sucrose when you're talking about the sugar that the phloem is transporting. Okay, so back to the mass flow theory. This has 3 elements. Number 1, transfer of sucrose into sieve elements from photosynthesizing tissues. Number 2, mass flow of sucrose through sieve tube elements. And number 3 is the transfer of sucrose from the sieve tube elements into storage or other sink cells. So this seems pretty complicated, but I'm going to go through each step nice and clearly. You might be looking at this thinking, what even is this sieve tube element that she's going on about? And so it's just a reminder that this is what phloem looks like. And that this here is the sieve tube element, which is just a long, thin structure arranged end to end. And this odd looking cell next to it is called the companion cell, because sieve tube elements are really basic and they can't keep themselves alive. And so they require a bit of extra help from this companion cell, which can respire for it and excrete products for it. So this means that there's pores between these 2 cells so that substances can pass between them. That should just be a really quick recap of phloem. So back to these key stages then. So as we know, sucrose is manufactured in cells with chloroplasts during photosynthesis. Now, because this cell is making lots of sugar during photosynthesis, there is a high concentration of sugar found in the source. Sucrose can now move from a high concentration at the source to a low concentration by facilitated diffusion into the companion cells. So sucrose is just moving down a concentration gradient. Now that sucrose is in the companion cells, it needs to move into the sieve tube of the phloem. This is done by active transport, and the textbook goes into quite a bit of detail how this happens, and it turns out that sucrose is actually co-transported, but you don't need to know that for the exam. All you need to know to get the marks is that sucrose moves from the companion cell into the sieve tubules by active transport. Now that sucrose is in the phloem, we go on to the second element of the mass flow theory. The presence of new sucrose in the sieve tube elements cause the phloem to have a lower water potential or more negative water potential. This is because we're adding more impurities to the phloem in the form of sucrose, and so the contents of the phloem is becoming less and less like pure water, aka a water potential closer to 0. So as the xylem, which is quite close to the phloem, has a really high water potential, one that's quite close to 0, water moves from the xylem into the sieve tubules just by basic osmosis. This creates a high hydrostatic pressure within the sieve tubes because we're adding water to the phloem, and so this pressure is really building up within the phloem. The phrase high hydrostatic pressure is really important to use when we're answering these exam questions. Just hydro meaning water, and we're really just building that pressure within the phloem. Now let's look at the respiring cells, or the sink. These cells are constantly using up sugars, either during respiration or converting it to starch for storage. These cells therefore have a very low sucrose content because they're constantly using it up. On the other hand, there is a high sucrose concentration in the sieve tube elements, and so sucrose moves into the respiring cells by facilitated diffusion from a high concentration to a low concentration. Because all this sucrose has moved into the respiring cells, there's now a low water potential there. And so water also moves into the respiring cells from the sieve tubes by osmosis. So, to summarise, in this step, sucrose and water are moving into the respiring cells, or the sink, by facilitated diffusion and osmosis, respectively. The next step explains how there is an overall mass flow. The hydrostatic pressure in this lower region of the diagram is lowered because water and sucrose has moved out of the phloem into the sink. However, like we said before, there is a high hydrostatic pressure at the top of this diagram because water enters from the phloem and sucrose enters from the source. So there is a high hydrostatic pressure at the source and a low hydrostatic pressure at the sink. There is therefore a mass flow of sucrose solution down this hydrostatic pressure gradient in the sieve tubules. And that's a really key phrasing that you need to use in an exam. Whilst mass flow is a passive process, it occurs as a result of the active transport of sugars, because if you remember in step 2, active transport is used to transport sucrose. The process, therefore, as a whole is active in that it requires ATP for this active transport. This is important to know because exam questions could ask you why the translocation of sugars is affected by temperature or metabolic poisons, and it's because of the need for ATP in the active transport of sucrose in step 2. So now let's look at an exam style question of describe the process involved in the transport of sugars in plant systems, and this is for 5 marks. So I'm just going to put the mark scheme up now. And I think the mark scheme really gives a nice summary of the process of mass transport in plants. So let's have a look at this. Obviously, you only need 5 full marks, but I would highly recommend learning all 6. So at the source of these photosynthesizing cells with chloroplasts, sucrose is actively transported into the phloem or sieve tube or element. So they're allowing you to have any of those. And then it goes on to say that this is done via companion cells. Note here that you don't have to specify that the initial process is facilitated diffusion, but I do think it's worth knowing just to help your understanding. This then lowers the water potential in the phloem and water enters by osmosis from the xylem, as we know. This produces high hydrostatic pressure. And then you can simply get a mark for stating that this is called mass flow towards the sink. Note that the word mass is underlined, and so you need to have this for the mark. You can't simply say flow or transport. And they do allow you to say either towards the sink, roots, or storage tissue. And then for the 6th mark, you just need to say what happens to the sugar at the sink. And you see there, it allows you to say that the sugars can be used in respiration. Like I've said, this mark scheme accepts sugar or sucrose, but does not accept glucose, so do make sure you remember that. I hope this was helpful in trying to understand the steps involved in the translocation of sugars in plants.